The very first stage of cellular respiration is called glycolysis. So, just like you should do with lots of biology words, you should break the word down as best you can. So, when we see glycolysis, we see glyco and we see lysis. So, remember, glyco is, I think in Greek it means sweet. So, glyco means sugar, right? Like glucose. And lysis means to split. So we're going to split sugar, and specifically we're going to split glucose. That's the sugar we're going to follow through. Glycolysis happens in the cytosol. It does not happen in the mitochondria. So interestingly, all organisms are able to do some form of glycolysis because it doesn't require a mitochondria at all, and it doesn't require oxygen. It's an anaerobic process, so oxygen is absent, as opposed to you and I, we're aerobic organisms. We require oxygen. So interestingly, this is a process that happens in all living organisms, and it's sort of an evolutionary connecting point for all organisms, because this is even the earliest organisms, 5 billion, not 5 billion, we haven't been around that long, 3 billion years ago, did some form of glycolysis. Simplest microorganisms did glycolysis. So it's pretty neat. And it only makes a very, very small amount of ATP, just 2 ATPs from glycolysis. So this stage is fine if you're a yeast and 1 cell big, or fine if you're a bacteria and you're just 1 cell big. Not good enough for us. We're 30 to 40 to 50 trillion cells big. So for every glucose molecule, if you're only making 2 ATP, it's just not enough to get by. So the next slide, you're going to watch a video. You're going to go back to our Moodle page and watch a video on glycolysis, and then come back. So you just watched the video on glycolysis. There's lots of videos out there on glycolysis. You can just search the web and you'll find all sorts of videos on glycolysis, but you just have to remember the big things, the big points here. I don't want you to get nitty gritty into the weeds on this. All I want you to remember is that we start with a glucose molecule, and that packs the most potential energy of all the molecules we're going to talk about. It makes sense. We're going to start with the one with the most energy, and we're going to strip it of its energy. There'd be no point in starting with something that has a little bit of energy. So we're going to start with the glucose molecule, and we're going to basically split it in half, right? If glucose is C6H12O6, so 6 carbons, we're going to break it in half and make 2 3-carbon molecules. So 2 ATPs have to be used in order to destabilize this molecule, because it's very stable. Glucose is very stable. It doesn't really want to break bonds. It doesn't want to steal electrons. It doesn't want to do any of that stuff. So we're going to add 2 ATPs to destabilize it. We call this the energy investment phase. So that kind of makes the molecule a little bit shaky and causes it to react. And then ultimately what we're going to do is we're going to make 4 ATPs from the process. Right? We're not going to get into the weeds on this. We're not going to get into every single little step. I just want you to get the big picture here. So 4 ATP molecules are going to be produced. Therefore, we have a net gain of 2 ATP, right? So we used 2, we gained 4, so that's a net of 2 ATP. So that's our energy payoff for this one, is 2 ATP. And remember, NADH is going to be our superstar for this whole process. So through the process of breaking or oxidizing that glucose molecule, breaking the bonds, releasing those hydrogens and electrons, NAD is going to pick them up and produce NADH. So we're going to make 2 NADHs from this process. Through the oxidation of glucose, we're going to make 2 NADHs. So NADH is just an NAD that has picked up a hydrogen and electrons and became NADH. And then ultimately what's going to happen is that glucose molecule is going to be broken in half and make 2 molecules of pyruvate. And I'm going to show you this in the next slide. So here in this slide, what we see, I'm going to get my little laser pointer again. We can see here is our glucose molecule. That glucose molecule is shown here with these 6 gray circles. And those gray circles represent the 6 carbons in glucose, the 6H12O6. So we're going to start out with our glucose molecule, and like I said, we don't have all the steps in here, we just have the final products. All that glucose is going to be destabilized, and we're going to break it in half, and we're going to end up with 2 3-carbon pyruvate molecules. This is a pyruvate, this is a pyruvate. So far you can see we didn't lose any carbon yet, right? All the carbons are still there. But what we did lose is some hydrogens, and those got picked up by NADH. So we made 2 NADHs here. We also made 2 ATP. So glycolysis, I always think of 2-2-2. Glycolysis, you make 2 ATP, you make 2 NADHs, and you make 2 pyruvates. All right, try the concept check again. Go back, go into your notes, try to remember what we just talked about. Answer these questions before you click to the next slide. I hope you did well on the concept check. Remember, if you have any questions, just save that for our Zoom meetings. So now we're going to move on to the next phase. So far we have taken that glucose molecule, split it into 2 pyruvate molecules. We've also made 2 ATP in glycolysis. Remember, that's not a lot of ATP for a glucose molecule, but something like a bacteria can utilize that very well. Some organisms just do glycolysis. We call it fermentation. And those organisms like lactobacillus bacteria that make yogurt, right? They take big vats of lactobacillus bacteria. They put it in with dairy, and the bacteria are going to take the sugars in the dairy and they're going to convert it into lactic acid, and that's what makes yogurt tangy. So other organisms will do the same thing, where you take a big vat of cabbage, let's say, and you put these bacteria in there, and they do anaerobic fermentation, and they're going to convert the sugars that are in that vat of, let's say, cabbage, and convert it into lactic acid, and that makes things like sauerkraut. Or even like yeast will do the same thing, and instead of converting that pyruvate into lactic acid, they convert the pyruvate into alcohol, and you get beer from that, or you get bread that rises, or dough that rises, really. And that's all from those organisms doing fermentation. So some organisms just stop at glycolysis, but for us, we are much, much larger. We're trillions of cells big, and we're aerobic. We use oxygen in the process of making energy. So since we have oxygen present, that pyruvate is going to get pulled into the mitochondria. So now everything is moving into the mitochondria because we're aerobic eukaryotes. And now we're going to start the next stages. So like I said, some books separate these 2 stages, some put them together. The first thing that's going to happen is we're going to prep the molecule for the Krebs cycle, and then we're going to do the Krebs cycle. So don't worry about the semantics of the names, but what we're basically going to do is we're going to prep the molecule of pyruvate and turn it into what's called acetyl. And then we're going to take that acetyl, and it's going to now, because we've kind of tweaked the molecule, we've actually taken a carbon off, that molecule now fits into the Krebs cycle, and we can do the Krebs cycle. Most importantly, what happens in the Krebs cycle is that it is what we call the final or oxidation of glucose. At the end of the Krebs cycle, there is no more glucose left. It's all gone. The whole entire glucose molecule has been oxidized, and there's nothing left. Okay, so that's important for you to notice. So once again, the next slide is for you to watch a video. So come back to me after the video. Okay, so you just watched a video on the Krebs cycle, and there's a slide after that, but I'm going to explain this with the image. I think it's easier. So remember, we got pyruvate from the stage called glycolysis, and now it is entering into the mitochondria because oxygen's present and it's pulling the electrons through. We'll cover the oxygen electrons at the end. And so what's going to happen, the first thing that's going to happen is this pyruvate is going to have an enzyme attached to it. And that enzyme is called a decarboxylase enzyme. Those are weeds, but I just said it because it's a good word and I like it. And I think that you guys are there and you can handle it. So this is a decarboxylase enzyme, ACE, right at the end. And it's going to break this bond right here. And it's going to release this carbon. And it's going to release the carbon and an oxygen, 2 oxygens, as carbon dioxide. So that's our very first carbon that we've lost. So now that carbon dioxide, I imagine you can probably guess where it goes. That carbon dioxide becomes the carbon dioxide that we breathe out every day, all day long, all night long, and all that stuff. All right, so that's the first thing that's going to happen. So remember, when we break this bond, we're also going to release hydrogens and electrons, and those are going to be picked up by NAD and make NADH, right? This is sort of our little reduction protector. It's going to pick up these electrons that are potentially dangerous to us and these hydrogen ions. Remember, a hydrogen ion is just a proton. And it's going to pick these up and it's going to eventually take them to the electron transport chain. We're going to see that next in the at the end, the last step. Well, now since we broke this carbon off, our pyruvate is no longer pyruvate. It has a new name, and its new name is acetyl. This 2-carbon molecule here is called acetyl, and acetyl is now ready to enter into the Krebs cycle. But acetyl needs sort of an usher, is what I always like to say, and coenzyme A is the little usher that allows for the acetyl molecule to fit into the Krebs cycle molecule. So there's enzymes at every step. So in order for this acetyl to fit into the next step, it needs a helper, and coenzyme A is the helper. So just to recap the prep stage, at the end of the prep stage, we make a carbon dioxide and we make an NADH and we have acetyl moving on. But one thing you have to remember, and I remember having a little bit of a challenging time with this as a student 127 years ago, we had to take 2 pyruvates that came to the prep stage for the Krebs cycle, right? I'm only showing one. We only show 1 pyruvate here, but in reality there are 2. So you have to multiply all of this by 2. So we really made 2 NADHs and 2 CO2s and 2 acetyls enter into the Krebs cycle. So I know that's a little bit challenging to say, but we have to make sure that we remember to multiply everything by 2 because there were 2 pyruvates that we made from this single glucose molecule that we made.